Okay, protein synthesis today. So this is all about how a sequence of bases in a gene gets made into a protein. So I've got a little picture of a cell here, and we can see there are two organelles in my cell, but actually what we've got to remember is that this cell has all the organelles that any other eukaryotic cell would have. It would have endoplasmic reticulum, Golgi bodies, maybe some lysosomes, definitely some mitochondria. But the only two organelles that I've drawn that are important for this lesson are the nucleus, because it contains the DNA, which has the genes that code for proteins, and a ribosome, which is where proteins are made, a site for protein synthesis. So this is my little diagram of DNA here. We can see it's double-stranded and sort of composes a little double helix. And we can see that I've highlighted one section of the DNA to show that that represents a gene. What I'd like to do before I go on is just have a quick reminder of what DNA and RNA are made up of. They're made up of building blocks called nucleotides. And nucleotides have three components: a phosphate, a sugar, and a base. And to make a DNA molecule or an RNA molecule, you have to join lots and lots of these nucleotides together. So I'm going to do a couple of little drawings later in this lesson, and I'm going to represent my nucleotides like this. Okay, so this little vertical line here is meant to represent the sugar and the phosphate, and then this little horizontal line here is meant to represent the base, sticking out at right angles. Okay, and if we have lots of these joined together, then you end up with a sort of line of nucleotides. If I just draw in another nucleotide and another nucleotide, and we can see that they make bonds with one another to join together. So that's one strand of nucleotides, and we know that DNA is made up of two strands, and so we'd have a strand that's joined to the other strand, and they are joined between the bases. So the bases are held together by hydrogen bonds, and they are held in place by the complementary base pairing rule, which we'll talk about in this lesson. Okay, so that's just a recap of the structure of this. This would be DNA, and we'll think about how RNA might be slightly different later on in this lesson. Okay, so let's get back to this original picture, which shows one gene, and if this gene wanted to be made into a protein at the ribosome, then the DNA molecule would have to unzip at the point where the gene is, and I'm going to draw it like this. So there's the double-stranded bit of DNA, and we can see that where it's unzipped, the bases on both of the strands are exposed. Now, one of these strands carries the code for the protein. So it's important that these bases here, which is going to carry the code for the protein, is exposed. So let's have a look at this process in a bit more detail in this space here. Okay, so let's draw the DNA unzipping and a little bit bigger. Okay, so there we are. So here's the double-stranded section. Remember, using the picture that I just showed you, these are the complementary bases holding the two strands together and the hydrogen bonds between the bases. And at this point, the hydrogen bonds have broken so that the strands can separate. And this is important because these bases here, and let's say that this is the coding strand, this is going to be the strand that carries the code for the protein. These bases here must be exposed, and we'll see why in a second. Okay, let's just draw in the other bases on the non-coding strand. They're not going to be copied because they don't code for a protein. Okay, so let's assign some names to these particular bases. Let's say that's, I don't know, T, A, C. So I'm naming these bases now. Thymine, adenine, cytosine. Should we have another cytosine and a guanine? Maybe another T and an A, and maybe another C. Okay, so we've got a particular sequence of bases which is going to code for our protein. Now, let's just go back to this picture here. We know that the DNA has to unzip. Let's just go back to this picture here. Why can't the DNA go to the ribosome and that sequence of bases be coding for a particular sequence of amino acids? Why can't that happen? Well, in order to get the code from there to there, the code has to fit through one of these little tiny nuclear pores, little gaps in the nuclear membrane. And the DNA molecule is huge. It carries hundreds of genes, not just one gene, hundreds of genes per chromosome, and it's far too large to be able to fit through the nuclear pore. So what has to happen is this code for one protein has to be copied, and then it's that copy of the code that leaves the nucleus and goes off to the ribosome. So let's see how that's copied then. We now have a series of RNA nucleotides. Okay, I'm going to draw them in a different colour. So these are RNA nucleotides. And what we're going to do is we're going to copy the code on the DNA coding strand using the RNA nucleotides, using the complementary base pairing rule. So let's just draw in the individual nucleotides, complementary base pairing. There we go. And now let's write in what these bases should be. So the bases in RNA are A, U, C, and G. Okay, so whereas DNA has A, T, C, and G, RNA doesn't have T, it has U instead. Okay, so using the complementary base pairing rule, T will complementary base pair with A, and RNA has A. So that base there would be A. A would complementary base pair with T if it was DNA, but because it's RNA, it will be U. C complementary base pairs with G. The next one is also G. G complementary base pairs with C, and T complementary base pairs with A. A complementary base pairs with T normally in DNA, but because it's RNA, it's U, and C complementary base pairs with G. Okay, so now we have the RNA nucleotides joined together. Well, I say they're actually not joined together yet, but they're all lined up in the correct order, depending on, determined by the code in the DNA. So something has to join them together. So something has to build that new sugar phosphate backbone. And the enzyme that does that is called RNA polymerase. That's really important. So RNA polymerase joins the RNA nucleotides together. And then what happens is the RNA now peels off from the DNA. The DNA closes back up again until it's needed. Okay. Meanwhile, the RNA has to finish off being processed. So let's have a look and see what we've got now. We have, and I've drawn it a lot longer than I did here just to demonstrate something. So what we've got first of all is the primary RNA. And actually we're going to call this now what it's meant to be called. It's primary mRNA. M for messenger. Okay, and if you look really carefully, we have sections within the mRNA. Some of them are useful coding sections called exons, and some of them are non-useful, non-coding sections called introns. So what we have to do is we need to get rid of the introns because they're non-coding. And the way that I remember this, these have to be removed. The way that I remember this is that the intron interrupts. And so that's why we've got to get rid of it. It interrupts the code. So our exon must be shunted together with the other exon and the intron must be taken out. And that process is called splicing. Let's just draw the finished product and then I'll show you something to kind of illustrate it. So if we just remove the introns, or the one intron in this case, we're basically shunting together the exon on the left with the exon on the right. This process is called splicing. Okay, let me show you what I mean. So I have here a piece of mRNA. Okay, there's the exon, there's the intron, there's another exon, and we want to get rid of that intron. So what we do is we just pull the right hand side of that exon to the left hand side of that exon. There we go. And you can see that the intron has kind of been pulled up into a loop. I've got a bit of sellotape here, so we're going to stick those together like that, and then that bit, that's the splicing bit. Just going to get rid of that. That's discarded and broken down, and we're left with our two exons joined together. Okay, and of course there's enzymes that are involved in doing that. Let's just write that down. That's called splicing. So the introns are spliced out, and what we now have is a mature mRNA. And that's what's going to leave the nucleus and go off to the ribosome and have its sequence of bases translated into a sequence of amino acids. So let's just finish this little mini tutorial. This first little mini tutorial was all to do with a process called transcription. Transcription is where you copy the DNA to mRNA, and it all happens inside the nucleus.